So if would you be able to introduce yourself? Um,
Sure,
Provide me with some background information, like what's your role at University of Vermont?
sure,
How
yeah,
long have you been working there, that sort of thing?
yeah, totally. I just got here about a little less, well, a year ago, almost exactly, and I'm a assistant professor in the Department of Surgery, and I study RNA biology for the most part. And I came from California about a year ago, working on RNA biology and immune cells. And so now that I'm here, I was kind of studying RNA and how it acts as a danger associated molecular pattern, or basically how RNA sensing works in the immune system. And COVID started and all the labs started shutting down. And I had a pretty close collaborator in California who is pretty awesome at RNA biology, and I think I'm pretty okay at it. And we here at the University of Vermont have a BSL-3 facility that allows us to study SARS coronavirus. Um, and so it just seemed like there were so many big questions regarding how how coronaviruses themselves um, take over the host cell. And we were reading papers from the 70s and 80s uh, to try to figure it out. But the truth is that a lot of the funding for basic coronavirus research has dried up at multiple points in the last, say, 30, 40 years. And so there are just big blind spots in how the virus works. And so me and my collaborator, Mitch Gutman at Caltech, uh, kind of just went through all the proteins in the coronavirus and tried to figure out where those proteins and were binding in if they were binding RNAs from the host. And then when we got a few hits, we followed up on those hits. So uh, basically, I came into UVM to work on RNA and the immune system, and because uh, the whole school kind of shut down during during the quarantine, um, it left me with a lot of curiosity and time to really work on this. And Vermont is pretty awesome because it has pretty awesome facilities for working on um, uh, virology, really.
Oh, that's really exciting. Cool. Okay, so you've been interested in RNA, and then this just perfect storm um, left you
Yeah,
with some
right.
curiosity.
Yeah, I mean, I think like when it comes to how the cell handles RNA and regulates mRNAs is something that is not terribly well understood uh, when it comes to immunology. There's a lot of room for what's called post-transcriptional regulation of transcripts after uh, production of the mRNA. And so it's kind of exciting to, like, there are just a lot of players involved, and there's a lot of differential regulation of how RNA is made. And that at the end of the day affects gene regulation and what transcripts are made and what cytokines are made. And it's really important, but I think it's quite understudied. And so so that's my focus.
Very cool. And so then this interest resulted in a pretty recent paper. I think October is when it was published in the journal Cell.
Right.
Um, and you found that the SARS-CoV-2 virus disrupts splicing, translation, and protein trafficking, and that's one of the ways that it um, suppresses the host cell's defense mechanisms. And I guess you pretty much already answered my next question, which was how did you come to investigate it? And so, can you give me like a little rundown about what you went in thinking you would find and what you ended up finding? Like, what was surprising?
Sure. Yeah. I
In
mean, I
general?
think we went in thinking that. So we went in basically. There are 27 proteins in the SARS proteome, and it's pretty compact, but it is one of the biggest RNA viruses there is. And biggest meaning it makes a lot of protein. So a lot of RNA viruses encode for like 4, 5, 6 different proteins. Flu encodes for 9 to 10, HIV, similar amounts. So here we have a lot of proteins, and we don't know what a lot of them do, which is super curious for us, because it's really important to know what they all do at this point. And it's kind of a black box for some of these, and they call them NSPs, non-structural proteins. And so we took every single one, and we basically used a cross-linker or used UV cross-linking to cross-link any RNA that these proteins were binding to the proteins. And most of them are all RNA binding proteins, because this is a virus that's an RNA virus. And so most of the proteins have to do with a lot of, RNA handling. Many of the proteins handle RNA, move RNA, reproduce, replicate RNA, cut up RNA, cap RNA. So a lot of the proteins do things that have to do with RNA. So we expected to find a lot of I thought that there would maybe be some interesting binding to 1 or 2, mRNAs that are really interesting. And in the end, we did find some mRNA binding, but there weren't any mRNAs that really stood out or made sense. What we did find is that a lot of the machinery in the cell, the ribosome, the spliceosome, and the signal recognition particle, which represents how mRNAs are shuttled from to ribosomes at the membrane. We found that each of these kind of machineries were targeted by different non-structural proteins, NSPs. And we tried everything. We looked at what all the we tried, so there are 27 proteins. We cross-linked every single one of them, but the non-structural proteins by and large had the most interesting binding profiles. And so what we think is going on here is that these proteins are able to shut down really important critical parts of how the cell works, so that the cell can focus on making more virus. And one of the ways to shut the cell off is to turn off the ribosome for anything that's not virus. And so this NSP1 is this really, really cool mechanism where it puts a cork into the ribosome so that protein can't be made, unless the RNA that's encoding the protein has a special sequence that says it's from the virus. So it's like a licensing mechanism for the ribosome. And similarly, we think we have the same kind of mechanism, or an interesting mechanism like that, for whether the protein can go into the membrane, so like membrane proteins or secreted proteins. And finally, we see similar things for the spliceosome, where it looks like the spliceosome machinery is just like blocked by coronavirus NSP16. So these sets of proteins seem to be actually shutting off big parts of how the cell works in order for the cell to focus only on making coronavirus particles.
When I read this article, I was like, wow, this really just amazing. It's does so many things at once, in like having that 5' leader segment to allow the RNAs to be made if they're from the virus. I don't know.
I'm totally on the outside. I'm not a virologist. So we worked with virologists, but I'm not a virologist. And so like when you're a virologist, you see all of these proteins as Swiss Army knives, and they do a million things, and like you can never make a point mutation because it might affect like 5 different functions of the protein. So for me, it was kind of like, seeing it being a tourist in virology, and just being like amazed at the multifunctional nature of each of these proteins. And really, I mean, they're the product of like intense amounts of evolution. So it stands to reason that they are multifunctional, and every amino acid matters. But to me, it's like it was the first time I had really dealt with this kind of thing.
Yeah, really, really neat. So I guess those are some of the details, and I think you touched on this a little bit, like what's the overall takeaway message? What do you want people to know about what you found? Yeah.
Yeah, I mean, I guess like the thing that the big picture goal is to understand what all of these kind of black box molecules are doing. And the reason why we want to do that, the big endeavor, is to say, we understand how all of the different subunits of SARS coronavirus do what they're doing in order for it to be such a weird and transmissible and infectious disease. And unless we understand what most of those 30 pieces are doing, we still are kind of at a loss for why there's, weird like numbness for some coronavirus patients, or why there's, extreme transmissibility. There are just things we don't understand about this virus that make it stand apart from other viruses and other coronaviruses even. And we are understandably just a little blind to the basic nature of coronaviruses in general. They're not studied very well. And so we know the basic stuff. We know that NSP12 is in charge of reproducing, of replicating the RNA. And we know that NSP14 and 16 are in charge of putting a cap on that RNA. So we know a lot of really basic things that have been elucidated over 20, 30 years. But there are probably, 3, 4, 5-fold more things that we just don't understand about how the virus replicates. And I think to really develop great antivirals, you kind of have to know what stealthy things the virus is actually doing. And I think we're at the point where we're describing the super first draft of what, maybe second draft, let's say all the people in the last 30 years got us to the first draft, and we're on the second or third draft of what this virus is doing. And it's incredibly more like magnificent and detailed than we even began to guess when we started, which is the case for all viruses. They do a lot more than you think they do when you put a magnifying glass on them. So so the big project is to come up with the next draft of what the coronavirus is actually doing, so that we can develop great antivirals and predict kind of why or how the virus will behave, as it continues to evolve and continues to be a public health problem.
Absolutely. Is that what you're going to be doing moving forward? Are you going to continue work on?
Yeah, so we're working now on kind of how the some of the weird intracellular features of SARS work. What happens is the RNA gets into the cell, the first bit of RNA that comes out of the virus, and then it forms these vesicles inside of the cytoplasm that are like protected castles that just are responsible for replicating RNA and keep all of the sensors, all of the immunity that the cell has, keeps all that stuff out and just makes lots and lots and lots of RNA without being detected. So I'm really interested in understanding the formation of those kind of protected, what they call double membrane vesicles. And so we're doing screens and trying to do a lot of biochemistry and RNA work to just piece out how that's working.
Is that unique to SARS-CoV-2?
It's unique to coronaviruses.
Okay. So interesting. Wow. Viruses are crazy.
They're fun. Yeah, it's really interesting.
I had one other curiosity question that I came up with while reading. Well, I guess I have two. Do you feel like with the knowledge you've gained through this paper, you have the ability to inform the development of antiviral drugs or vaccines or such?
We're actively trying to find molecules that inhibit some of these NSPs. And so now that we know what some of them do, we have very cute, easy in vitro systems to test whether NSP1 is regulating translation. And so we're going through libraries of small molecules that are, you know, tens of millions big to try to find small things that are basically drugs that are able to kill the NSP1 activity. And this is how you do it. And so we're first, you got to figure out what they do, then you got to figure out things that block them. And so we're in the figuring out things that block them phase.
Very cool. And my last question, in terms of like this interview, would be reading through your paper. It seems like you had a really good system going where you like tested very specifically where does this bind, using various techniques depending on what you were looking at, like getting into the specifics, I guess the in vivo or the in vitro stuff. And then from there, you went and looked at like functionality specifically, and then globally, and then in infected cells infected with SARS-CoV-2. And then from there, you went and looked at the impact it had on the interferon response. And so it seems like you had this really good system. Do you feel like the science world might be developing a pandemic research model that might allow for this sort of research to happen more quickly? We keep hearing like there might be another, pandemic in the future if we don't change some of the ways we're doing things. And do you feel like there's been progress made in the ability to execute this research like in a smart, quicker way? If that makes any sense.
No, it does. It does. I want to give you a really hopeful answer, but I suspect not, honestly. I suspect that what was doable in the time that it was doable happened because, not just by us, but by the whole field, happened because you did have a lot of people that, really slummed it for 3 decades to try to get, work out. And it's really impressive. It's really, I mean, those guys, they worked really hard, and it was hard to get grants for something like coronavirus. I was talking to one person who worked on, the original mouse coronaviruses that were identified in the 60s. And he was like, they would like sleep in tents when they visited each other. There were only like 20 or 30 labs that were doing this in the United States, and it was really hard to be a coronavirus lab. And anyhow, so a lot of the work we did happened because of them, but funding levels for coronavirus will probably go up. But there are, I would say, dozens, if not hundreds of viruses that are looming threats. And so we have to be super proactive about like studying things that are not threats yet. But like there's a really seminal paper from 2015 from one of the big coronavirus labs. A SARS-like cluster of circulating bat coronaviruses shows potential for human emergence. And so like that was published in a really big science journal, but like it's not, virologists are, I just sent the title to you, but virologists, they'll say things like this, but it takes policymakers and Washington, D.C. in general to say, okay, well, maybe we should study some of these viruses that no one's working on. And so one of these older guys that worked on coronavirus forever said the only people that really cared about coronavirus work back in the day was that people who worked on cows, because a lot of, agriculture, pigs and cows got coronaviruses. And so there's a great coronavirus vaccine for domesticated animals, cats. But there's not such a thing for humans, and so we need to take a broader view on this. And I have a few grants out, and I have no idea what the coming trade winds will be in terms of fundability for rare viruses. But if 2005 there was a coronavirus pandemic, SARS-1 basically in 2002. There was a huge funding cliff that basically killed all coronavirus research, a lot of it, almost all of it. circa 2008, 2009. So if that happens again, then you just lose a lot of people that are interested in working on this kind of thing. And more importantly, you lose that level of interest of knowledge about something that could potentially pose, could potentially close down the United States for a few months. So I am not optimistic that something has fundamentally changed. I hope something has, and I think it will take courage and the wisdom of policymakers to get to the point where they realize that putting in the resources for viruses that have not yet posed a threat might really change the world.
Absolutely. Yeah, funding is huge.
And it's not a selfish thing. It's not for me. It's just there's a million virologists who many tens of thousands of virologists in the United States who will all tell you that it's really, really hard to be a virologist in the United States. Maybe not anymore. Hopefully not anymore, but it's hard.
Yeah, absolutely. I definitely listened to a couple interviews on this podcast that I like called The Drive. It's by Peter Attia. I don't know. He's had a lot of people on.
Yeah, I listened to it. It's great.
Yeah, love that podcast. But they've all brought up like,
Funding.
yeah, we've been trying, but
Yeah.
the funding has just been pretty bad, and yeah, but
Totally.
hopefully that will change because maybe we've learned our lesson, but I don't know.
Time will tell.
Time will tell. Okay, well, I'll let you go. Thank you so much for your time.
Cool. No, this was great.
It was
Have a
fun.
wonderful day. And good luck with the whole project.
Thank you so much.
Okay. All right. Bye-bye.
Bye.